Well. That was thank you very, very much, Girish, and I'd like to uh, thank the organizers, particularly Girish, for inviting me to give this presentation today. Listening to this morning's presentations, I'm a bit daunted and honored to contribute at this on this stage. I actually visited TMH now more than 30 years ago. Uh, and I remember meeting Raj Badwa then, Dr. Raj Badwa then. We were a year or two younger then, of course. And to see the development, the changes, the quality, and the quality of the work and the quality of the discussion is just so impressive. So it's really good to see that change over that period of time. I've been asked to speak to you about improving health faster by transforming clinical trials. So I'll start my presentation now. So where we might start this discussion is to think a little bit about what we currently do. And if you think about what we currently do in trials is we ask the question, does this new intervention improve outcomes for this specific disease? And if you speak to people around the world, you speak particularly to companies, they say this process can take up to 20 years, cost up to some £2 billion, $2 billion. And of course, what we've done for many of our diseases, this model has failed. What we've got is little or no progress. And I've just given you some diseases that I'm aware of that we've made little or no progress in for decades. For those of you involved in oncology, there's a number of cancers there. But there are many other diseases like motor neuron disease, dementia, Parkinson's disease, psychosis, whole host of diseases we've made little or no progress in improving outcomes for absolutely decades. And so that model that we have currently is not moving things along for a large number of our diseases. And if you look a little bit under the bonnet of this problem a bit more, well, the basic problem has been is not that we've been short of signals for new interventions for our diseases. In our phase 2 trials, of many phase 2 trials done around the world, we have signals of activity, and we have loads of them in the literature. What has typically then happened is those phase 2 signals have not translated into improvements in phase 3, into outcomes, clinical outcomes in the phase 3 trials. And actually, if you look at the literature, you'll find that across a wide range of diseases, most phase 3 trials do not show superiority of the new intervention over the old intervention. And added to that problem, so we're failing late in the day, added to that problem, that the phase 3 trial, if you talk to industry, costs about 75% of the evaluation of a new intervention. So that's where all the money goes. And of course, those of us who do these things know that they are much harder to do. They cost many millions of pounds or hundreds of millions of pounds if you're working with industry. You have many hundreds of individuals or participants in the study, or sometimes many thousands, and many sites, many multi-site, and many years. And often these are not just multi-site, multi-country as well. So these are big things to do. And a good example, I'll take you out of those who are on the cancer patch, I'll take you out of your cancer comfort zone here, because you can learn a lot from other diseases. I'll take the example of Parkinson's disease. This is a disease that's increasing worldwide, and we know some of the symptoms of Parkinson's disease. Horrible disease. And what I'm showing you here is a graphic which shows you in the brown boxes the phase 2 trials, the dots taking you across time, and the yellow box is showing you the phase 3 trials. So many phase 2 trials showing you signals of activity. Subsequently, at a period of time after that, the phase 3 trial has been done, and we've failed to show that the new intervention is better than the old. And we know that we've got only symptomatic treatments for Parkinson's disease. There are no disease-modifying therapies at all for Parkinson's disease, despite decades of research and absolutely billions of pounds and dollars being spent in that area. So how can we change our thinking in this area? Well, actually, you can do this really quite simply. If you change the thinking from the question, just change the question that we're asking. Does this specific, the question we currently ask is, does this specific intervention improve outcomes for this disease? That's what the phase 3 trial does. If we change that question to, how do we improve outcomes for this disease as quickly as possible, you come up with quite a different way of thinking. And if you think about it, actually the answer to that is really quite simple. If you're thinking of the clinical trial space, how do we improve outcomes as quickly as possible? Well, the first thing you do is you evaluate many interventions all in one protocol. Why wouldn't you do that? There's nothing magical about one intervention in one protocol at any given time. You focus on the phase 3 stage because that's where we keep failing, and that's where we spend all the money and all the time. We learn that in a number of our diseases, we acknowledge that many interventions will not succeed. We just know that. We just don't know which ones they're going to be. And we adapt the protocol as we go from data that's emerging from within the trial that you're doing within the protocol and from data that's emerging outside of the trial. Very simple concepts. And this leads to the concept of these platform protocols. We call them multi-arm, multi-stage platform trials, but there are also other types of platform protocols, basket trials, umbrella trials. But the principles are very, very simple. Evaluate many things, focus on the phase 3 stage, acknowledge that you're going to, many will fail, and adapt as we go. Very, very simple. And this is a schema just showing you that sort of idea. Traditional approach is shown at the top. We've got a control treatment against treatment A, a control treatment, and then that fails, treatment A fails. We go on test treatment B against control, that fails, and then we go to control. This is what we've done for decades. You can see immediately that if you actually just put them all together and have treatment A, treatment B, treatment C, all starting at the same time with one control group in one protocol, and at a point in time when treatment D comes along to be evaluated, enter that into the same protocol. You can see that's just going to be more efficient. It's a barn door obvious that it's more efficient. And these are what we call multi-arm, multi-stage platform trials. These platform protocols are becoming increasingly common now. Many have been developed, and most of the methodology have been developed for phase 2 trials, and most of the methods and most of the application has been done in phase 3 trials, phase 2 trials rather. And that, my own view is that's largely going to be a waste of time. It will move us forward a little bit, but not much, because that's not where we've had the problem. And a huge number of methods have been developed. They'll have limited impact because the problem has been failure at phase 3, not failure at phase 2. We don't need to solve the phase 2 problem. And so the huge amount of work going on in that area is led and is going to improve things a bit, but not very much. We need a sea change. Also, it's useful to note that organizations like yourselves and ourselves in the UK, the MRC, and other academic organizations have a unique place in this research environment, because these platform trials are uniquely going to be done, almost uniquely, not completely, almost uniquely going to be done by academia, because industry at any given time for any given disease will only have one intervention that they're interested in. And research history shows this, experience shows this, that academia is uniquely placed to do these trials. And as I say, unfortunately, most applications have been phase 2. That's partly because we have a tendency in society as a whole to do things we can do rather than things we should be doing. And we can do the phase 2 trials. The phase 3 trials like these are really hard to do, but as I'll show you, the prize is huge. These sorts of designs are faster. They give you multiple treatments tested at the same time. They're cheaper. They're much more efficient. They facilitate recruitment. These trials that we're doing of these sorts of designs have been the fastest recruiting trials we've ever done, and we can discuss perhaps why that's the case. And they're flexible. We can drop and add treatments to these. And I'll give you, and just before I give you a specific example, just give you a little bit of a history component to this. The first formal MAMS platform trials in the modern era, and I'll pick up why that's in the modern era in a moment, is our trial in prostate cancer, which is STAMPEDE, which I'll give you a sense of in a moment. We launched the trial in 2005. The first discussion about adding arms was in 2007. More formally into a platform trial in 2010. And over time, we've added 5 new research arms. So 10 research arms have been evaluated. And I'll give you a bit more depth on that in a bit. At the same time, colleagues in the States at the Berry Group developed the I-SPY trial, but that was a randomized phase 2 platform looking at many new drugs in breast cancer, particularly, launched in 2010. So this sort of started around late 2007, 2008, 2009, around there in the UK and in the US. Let me give you an example of this to make it real for you. When we started this thinking in about 2000, there are about just over 1 million new prostate cancers worldwide. In 2005, when we started launching STAMPEDE, treatment for high-risk prostate cancer had not changed for 40 years. Nothing had changed. Hormone therapy was the standard of care, and it was, I mean, how could that have been? We'd spent 40, 50 years getting nowhere. Median survival was around 3 years for men with metastatic disease. So we launched this trial in prostate cancer, phase 3 platform trial, and we've evaluated in that trial a chemotherapy agent, a bisphosphonate, a COX-2 inhibitor, a radiotherapy, and the new androgen receptor signaling inhibitors, metformin, which is a repurposed drug, and estrogens, hormone replacement therapy that was used in women, used for men, estrogens in a wide variety of treatments. And that's an important part of the design of these things, because we know that most of our diseases, if you're going to treat them effectively, you're going to need combination treatment. So you test a wide variety of different interventions and then combine them should they be successful. What we've managed to do since 2005 to 2025 is do 10 phase 3 comparisons in 20 years in one protocol. A mixture of old drugs, new drugs, repurposed agents, and radiotherapy, all in the same protocol. So this is the trial, and it's quite simple to follow the schema here. Across the top, you've got the standard of care going from 2005 to 2025, and then the 10 research arms that we've added over time. The block shows you the accrual period. The dotted line shows you the follow-up period, and the black box at the end tells you the time point of analysis for that. And you can see there's all of the arms. We've started 5 research arms, added a further 5 research arms over time, and analyzed those at various times. So this slide summarizes here, let's have a look, the basic outcomes or outputs from this trial. We've randomized 12,000 participants in this trial over 120 hospitals. As a consequence of the results of STAMPEDE and other trials which have done the same comparisons, we've changed standard of care 5 times within the trial. So every time we get a result, positive, we change the standard of care, add that standard of care to the control arm and to the research arm. Survival now, as a consequence of STAMPEDE and other trials which have contributed, we've improved survival for men with metastatic prostate cancer, a median of 3 years now to 7 years. And if you think of any other way you're going to try and do that in a 20-year period, evaluate 10 different types of therapy, improve median survival from 3 to 7 years, you cannot think of another way you're going to do that in that period. It probably would have taken you 2 generations to have done all those comparisons. All the stopping and starting, all the initiation of all those protocols. And these things are very flexible. You can drop research arms, add research arms, change the control in research arms from using the data from both within and outside of the trial. You can actually even change the primary outcome measure and the sample size, etc. very adaptive. This graph shows you on the left-hand side all the methodological publications that we've had out of this sort of design, because the design looks really quite simple, but actually underneath it, once you start opening up the methodological aspects, the methodology gets quite complex. It's very doable, but it gets complicated. But as I've shown you, the consequences of doing this sort of design are huge. And on the right-hand side, now, when I did this slide, more than 80 randomized trials have used this sort of design, particularly down here in the COVID era. A lot of the COVID trials use this sort of design. We're developing this sort of thinking going further. A number of you will be surgeons in this audience, and you'll know that wound infection after any type of surgery is one of the most common infections that you can get worldwide, of course, because there are so many surgical operations done. It's a major issue for the patient. It's a major issue for health services. And actually, we don't know very many ways of reducing wound infection rates. There's little progress been made. And the guidance on this is very, very poor. There's a poor evidence base. There's only generic guidance available on this. So what we've set up is this Rossini basket platform trial. It's multiple parallel, separately powered, multi-arm, multi-stage trials across specific body types, all in one protocol. For those who are more methodologically inclined, this is a basket factorial MAMS platform design. But you can see what it is here by this graphic here. So we've got 6 different types of surgery: breast, vascular, cardiac surgery, neurosurgery, amputations, and thoracic surgery. And a number of different interventions being evaluated in each of those surgical settings. Some of those interventions will be common across different surgical settings. And of course, at various points in time, if you find a successful intervention for one surgical setting, you might move it to another surgical setting. So this is just a very efficient way of doing lots of trials, evaluating lots of different interventions for different types of surgery, and learning both within each surgical intervention, but across surgical interventions. And also, for example, here, we know that the neurosurgery area will have probably fewer operations than, for example, the breast or the gynae one will. We know that. Well, can we use data from these more common surgical types to supplement the data for neurosurgery, because we'll never get enough patients in the neurosurgical setting. That doesn't mean to say we shouldn't investigate it, of course. Can we use those data to support that when the intervention is the same? The total sample size of this, we've designed this. Now, this trial is about to start. It's about 26,000 patients. But you can see over a period of next 5 to 10 years, we will make significant progress in this area with a little bit of a good wind and identifying appropriate interventions which might work. We're working, this sort of technology can be applied across a lot of areas. And I was just showing you the different areas that we're applying this sort of thinking in across many different diseases there that we're working on. It's not just us now working on this in this sort of design. The obvious question comes is, when should you prioritize such a strategy? And if you think about this superficially, you could argue that this could be when there's, these are big things. So you want lots of patients, you want short-term outcomes so you can make decisions quickly, and you want large funding pots. But actually, it's exactly the opposite. Because these things are much more efficient, these methodologies are much more efficient. If you haven't got many patients available, if the outcomes are long-term and you haven't got a huge amount of funding, this is exactly the sort of situation where you should be doing this design, because it's more efficient. And if you're going to make progress in our lifetimes in some of these diseases, you're going to have to change the way we think. So when should you prioritize such a strategy? Well, actually, I think Those areas which really require this sort of different type of thinking is where progress has been slow. And we know some of those diseases. I've listed some for you there. You will have your own list there. When a number of interventions with different targets are available, because we know that if we're going to make significant progress for many of our diseases, it's going to require combination therapy, not single agent therapy. You've got to have enough patients and enough doctors and researchers on board. And of course, the funders have got to be on board, because the nature of the funding envelope you need for these things is different to the standard one. I'll just end to conclude by saying, in a range of diseases, we need to take a different approach. If we carry on doing what we've been doing for years, we'll keep getting the same answers, and we'll keep making no or little progress. Part of this is planning for a 10 to 20-year horizon, focusing on the phase 3 evaluation. And this is a call for researchers and funders to change their strategies in a number of these diseases. And what we've found actually is key to this change has been the patient and public voice. Because my patient partner has asked me, when they've seen this sort of presentation, why don't you do all of your trials like this? And if anybody has a good answer to that question, I'd be very, very keen to hear it. And I'll stop there. Thank you very much.
That was simply a remarkable, very thought-provoking keynote. And I would say it was a perfect complement for the oration, where we talked of equity in healthcare, and now we are talking of pragmatism in healthcare, effectiveness, I would say a certain level of disruption, which is very much needed. So thank you so much for that. And to recognize the contribution, I invite Dr. Mohanty, Dr. Sudeep Gupta, Dr. Ramesh, and Dr. Chaturvedi to present the plaque to Professor Parmar. I would invite Dr. Mohanty and Dr. Sudeep Gupta to also come on stage to present the plaque.